Welcome everyone to the second video on enzymes. The first one was about digestion and food, and this one is more about enzymes, what they're made of, what they do, and tackling those 2 and 3 mark questions where you tend to get 1 mark but miss the others. So this is suitable for all, combined, and triple. Okay, so let's get into it. So our first thing is what a catalyst is. So if you are asked what is an enzyme, you need to say an enzyme is a biological catalyst. Now you would have done catalysts in chemistry. They speed up a reaction. Enzymes are exactly the same, they just happen to be biological because they're in the body. Okay, so an enzyme is a biological catalyst. Now that is fine if it's 1 mark, but if it's 2, which it sometimes is, you can also say they break down food from large molecules into smaller molecules. And if you are Edexcel, large insoluble molecules into smaller soluble molecules. They like those words as well. Okay, so if you're Edexcel, add those soluble and insoluble. Okay, so that's our definition. So when we eat food, our food would eventually get broken down, but there's not time between it going in this end and coming out that end. So we need these enzymes there to speed up that breaking down so we can actually absorb those food particles. Otherwise, we can't absorb the larger food particles. So that's our definition. Okay, people often get confused because enzymes are actually made of proteins, but also enzymes can break down proteins. So we get a little bit confused. So let's go 1 step at a time. Enzymes are protein molecules. So they're made of proteins, but not the same proteins they break down. And proteins are a long chain of amino acids. So enzymes, therefore, are made of long chains of amino acids. Now, if we look a little bit closer, those long chains of amino acids are folded into a particular structure to make a particular enzyme. So here we've got, over here we've got our chain here, and then this is representing our enzyme. Now obviously they don't look like this in real life, but this is a representation of these folded up amino acid chains into this protein. Now, as we know, each enzyme has its own shape around here, and that shape is determined by which amino acids are in the chain. So the enzyme that breaks down carbohydrates might have a different amino acid chain than the enzyme that breaks down proteins, and therefore they'll have a slightly different shape here. Hope that makes sense. So different types of enzymes have different shapes and functions because the sequence and type of amino acids is different. And all proteins in your body, there's so many, but just a few to name, like hemoglobin in your red blood cells, that's a protein. It's a folded up amino acid chain. Okay, so you've got lots and lots in your body. This is just some we're talking about here. So why are enzymes so specific? In other words, why do certain enzymes only break down proteins or only break down carbohydrates? It's because of how they fit together. So this is a representation over here of an enzyme. Now this is a 3D model, and this is a representation of the substrate or the food. Okay, Posh name, substrate. Just make sure that's a B there, substrate. Okay. So this piece of food will fit directly into this enzyme, and this bit is the active site. The bit that is the same shape as this, they will fit in. Okay, so different enzymes have different shapes, so they will fit different substrates or different food molecules. So let's just pop that in words that are easier to understand. Different enzymes have different, we're getting a bit low there, shape, active sites. So only one substrate, one food molecule will fit. And that's why we say enzymes are specific. So you couldn't take hemoglobin out of your blood cells and expect it to break down food, because it's a different type of protein, and so are these enzymes. All right, let's go on to the next bit. So this is the generalized picture of the lock and key hypothesis. Sometimes it's called lock and key method, lock and key hypothesis, and let's just talk through it, because sometimes you have to explain it. If you do have to explain it, you are allowed to draw a little diagram as long as it's labelled. Okay, you can draw a diagram as long as it's labelled, because it's a bit hard to explain this without a diagram. I think I would find that quite hard. So what we've got over here, this is our enzyme, and this is our substrate or our food, whatever that may be. And the enzyme and the substrate fit together really nicely, and that is called an enzyme substrate. So the two things that have locked together, complex. If you can't remember that, you can literally say the enzyme and the substrate. So you can say the substrate fits perfectly within the enzyme. If you can't remember enzyme substrate complex, as long as you're saying what happens. And then here's our enzyme again over here. Sorry, here. But what's happened is this substrate has broken now into two pieces. So that could have been a piece of carbohydrate, and it's broken down into glucose molecules. Okay, there's a little bit on the bottom here. So we've got the enzymes and reactant. I prefer substrate, so I would use substrate here. Enzyme substrate complex. These are not incorrect, it's just this flows better. And then we've got our enzymes and our products over here. So one of the questions at the end is about the lock and key. So I'll show you an example of how you would have to write your answer. Right, so that's what happens at the active site. Now the active site is very sensitive. So this bit here is very sensitive. So if this enzyme gets too hot, the active site will change shape. And I'm hoping you know that word. We can't say the enzyme dies because it's not alive, but we can say it is denatured, or it loses, the active site loses its shape. That's when it gets too hot. When it gets in the wrong pH, the active site will change shape. Okay, so it won't work so well. And enzymes don't denature till they're over about 60 or 70 degrees. So they'll work quite well up till then, and then they'll stop working if it gets too hot. Okay. Now, this is a little bit which comes up sometimes as a 1 marker, sometimes a 2 marker. More in Edexcel, but I have seen it come up in AQA, so just be aware. Excuse me. Just like catalysts in chemistry reduce the activation energy, of a reaction, so it goes faster. Enzymes do exactly the same. Okay, so here we go. So normally this top one here would be the activation energy. But what enzymes do is reduce it, so they reduce it by that much. So the reaction happens quicker, more quickly, I think is the correct term. So in terms of enzymes that are digesting food, this could be a carbohydrate. This enzyme could be amylase. And then the product could be glucose. All right, so if it says how does an enzyme work, or how does an enzyme speed up a reaction, reduces the activation energy. Okay, well done, good, good. Right, now along with your traditional enzyme experiments, excuse me, there's another one that comes up. Now, I don't know why examiners are so keen on this, but it's one that is an enzyme that breaks down food. It's a different enzyme in your body. And it's an enzyme in your body that is in your liver. Okay? And it breaks down this substance here, hydrogen peroxide. Now, we produce hydrogen peroxide from various metabolic reactions, but it's actually quite poisonous. So we need actually to break it down. And the liver is really good at doing this. Now, I think the reason examiners bring it up is because it's one experiment that you can actually see an enzyme working. You can't see an enzyme working in your stomach or anything. But this one, if you've got a bit of liver and a bit of hydrogen peroxide, you can see it working. So this hydrogen peroxide gets broken down into two harmless things, water and oxygen. Okay? Because if you look at H2O2, it's very similar to the water, but just that extra oxygen makes it a lot more toxic for our bodies. Right, there you go, water, oxygen. Now this enzyme is actually called catalase, and it's here. And it breaks down this hydrogen peroxide in the liver into water and oxygen. Now, sometimes these are plant questions as well, because plants produce catalase too. So don't be surprised if you get a plant question. Now, there is a short video here, which I'm hoping the link will work. So it's just showing you dropping a bit of liver into hydrogen peroxide and how it bubbles and oxygen is produced. Okay, so if the link works, please watch it. Perhaps one of you could let me know in the comments afterwards if it works. So it's one of the few reactions we can actually see, so it's quite good. It's just like a 5 to 10 second video, that's all. Right, so if you see catalase in a question, don't panic. Okay, you know it's an enzyme because it ends in A's, A-S-E, and all the enzymes end in A-S-E for GCSE. And it should tell you the reaction. It will show you the equation for the reaction, and then it'll ask you about other things about enzymes, which it always does. Okay, so don't be phased if you see that one. Right, if you want to do this, that's brilliant. Pause the video. This is the answer. What would the question be? Complete this for each key word. Okay, pause the video now if you want to have a go. Right, hopefully you've had a go. So amino acids. The question would be, where is, oh, it's up there, look. What are proteins made of, or something like that? What are proteins made of? Whoops. Catalyst. What speeds up a reaction or what reduces activation energy? Another one. Keep pressing that button. Okay. Enzyme. What is a biological catalyst called? Active site. Where does the substrate bind on the enzyme? You also don't have to be word for word, I'm just giving you ideas. Activation energy. What is the energy called that starts, a reaction? Okay, so you've had a good go. So well done, give yourself a nice mark there out of 6. Right, few questions. First one's a bit easier, the second one's a bit harder. So pause the video, have a little go at these if you want to, or you can do them with me. So the first one, what is an enzyme? Draw a ring around the correct answer. I'm hoping you all put catalyst. Well done. All enzymes are made of the same type of substance. What is this substance? Hopefully, I've drummed it into you. It's a protein. Where is the enzyme amylase produced in the body? Well, it's produced in 2 places, but it is produced in the salivary glands and it's also produced in the small intestine, but that's not on our list, so we can only have one. Stomach is proteases. Liver doesn't produce amylase. Okay, so hopefully you got all those 2 right. So they're going to get a little bit harder now. DNA can code for the amino acids of the active site of an enzyme. Explain the role of the active site of an enzyme. So pause the video. It's 2 marks. Think about what key words you might need for this one. Right, hopefully you've had a go. So I have got the mark scheme here. So we need to say something like it's a specific shape where it binds to the substrate or where the reaction takes place or it's catalyzed. They join together to break down molecules. Or you could mention the lock and key hypothesis. Now you only need 2 of these. Okay, so that's quite a big selection, isn't it? So you could say something like, the active site has a specific shape for the reaction to be catalyzed. Or the active site has a specific shape where it can join with substrate to break down molecules. Okay? Right, well done if you got 2 of those. Just remember, biology words get biology prizes. So, you know, it's all about those key words. Right, let's have a look at the next one. Right, 3 marks this one. Explain how the lock and key hypothesis models, explain how the lock and key hypothesis models, I think that's to say, show how enzymes work. You may use labeled diagrams in your answer. So remember me saying earlier, if you do a little labeled diagram, as long as it's labeled, then you can get the marks. Okay? So pause the video now, have a little go, either drawing it or explaining it, it's fine. And then we'll have a look at the answer. Right, well done. So hopefully you've got an answer now. You've formulated your answer. It's an explain, so we're going to need biology words. So if you haven't got biology words in there, pause again and have another go, but I'm sure you all have. Right, let's have a look at the mark scheme. Right, so we've got all this here, but you could have done it a lot easier. So let's do it in pictures first and then we'll look at the mark scheme. You could have nicely drawn, I don't know if I can draw it with this pen, but we'll have a go. you're labelled, so something like this. So enzyme, substrate. Then we could have had them together. Enzyme, substrate, complex. And this hasn't changed shape at all, but this bit has been broken down to smaller bits. Okay, substrate, broken down or catalyzed. Now, that is an awful lot of information, an awful lot of biology words. If you wanted to make doubly short, you could call this 1, 2, and 3. And you could put it in words as well. So let's have a look at the word answers. Enzyme and substrate have complementary shapes. This may be awarded if clearly shown in an unlabelled diagram. So if you've labelled your diagram, you're going to get it, okay? Because complementary means they fit perfectly together. Not you've got a nice pair of trousers. That's complementary with an I in the middle. Complementary, things fitting together perfectly. Substrate fits into enzymes or enzyme substrate complex forms. Well, you put that on our diagram, didn't we? Reference to active site. So if you're doing it in words or on your diagram, you could have shown where the active site was. Enzymes break bonds, or reactions occur or make products, or the idea of products leaving the enzyme so that the enzyme can be used again. Now, a word about mark schemes. If it's in brackets, you don't need to have it. It's just there to make a little bit more sense. So even if you just put products leave enzyme, or you show it on your diagram, you've got the mark. Okay, so we needed 3 of these. So I'm really, really, really hoping that you all got 3 on there. And I'm going to upload the enzyme required practical soon. So hopefully you'll have that to look at as well. Right, happy revising everyone. You're doing a brilliant job and I will see you in the next one. Take care.